Welcome back to another AP Biology video. In this video, we are going to dive into the next part of what we call the central dogma of molecular biology, and that is the formation of an RNA transcript from our DNA and the subsequent processing that it receives in order to be eventually producing a protein. So if you guys are ready, we will jump right into it. So starting off, when we use the term central dogma, we're referring to this unidirectional flow. So it flows in one way from biochemical information. So we go from the language of nucleic acids, that is DNA, to the language of proteins, which are amino acids. And this process includes two separate events or mechanisms that occur within the cell. These include transcription, which is going to be the focus of our video today, which is a process that builds mRNA using DNA as its coding template. And then in the next video, we're going to look at the process of translation, where we build these amino acid chains that will eventually be our proteins using messages that are carried on that RNA molecule. Altogether, these two processes are sometimes referred to as gene expression, hence the name of our unit this go around. What's important to note is that we will touch in the next video on the fact that while we call this a unidirectional flow, we can actually reverse transcribe going from mRNA to our DNA, but that is the only part of the process that we can kind of reverse engineer one form of nucleic acids to another. So we're staying within the same language. We can't, however, go from protein to mRNA. So if you guys are ready, we are going to look at all of the great details of how we are able to produce an mRNA transcript from a DNA molecule that is held within our nucleus. So, first, let's establish that a gene is this sequence of DNA that is used to encode a protein. You have all sorts of genes for a variety of different characteristics in your genome. When we undergo the process of transcription, we are going to use one primary enzyme. This process is a lot simpler than what we looked at in the previous video with DNA replication, and this is RNA polymerase. So just like DNA polymerase built DNA strands, RNA polymerase builds RNA strands. And so we are going to catalyze this synthesis in a 3 to 5' direction. And this is all going to be dependent on which part of the DNA we are reading as our coding strand and which one is our template strand. So these are some terms to keep in mind. Now, RNA polymerase knows attach to a particular gene by looking for a region that we call a promoter. This is where transcription begins. This is where RNA polymerase is going to attach to the gene sequence. And so we have within that promoter what we call an initiation site, and then we can describe DNA going in either direction. So if we are downstream of our initiation site, these are genes that are not going to be transcribed. If we are upstream, we are genes that are going to be transcribed at least until we reach a terminator. So to be terminated means to end. So the terminator sequence is the portion of our gene where we tell RNA polymerase, hey, look, it's time to stop. We're ready to wrap up this process. So when we look at the DNA strands that we have starting out in our nucleus, we can define them based on the role that they are playing in the process of transcription. So we will have a coding strand and we will have a template strand. The coding strand is the one that contains the code that we are looking for, that we want to make a copy of. And the template strand is going to be complementary to that coding strand. So that when we want to build a version of the coding strand, we want to use the complementary version of it so that we can match each of those complementary pairs and bases and get a nice, usable, workable copy. So you'll notice that our RNA molecule that we're producing from this strand is the same sequence as our coding strand, except for one major difference. Because this is RNA and not DNA, we have to take out all of those thymine molecules and replace them with uracil. mRNA strand itself is built in a 5' to 3' direction by pairing these free RNA nucleotides from the surrounding environment to the DNA template. And so we are going to form these bonds according to our complementary base pairing rules. So that is T with A with U, G with C, and C with G. So most of those, at least half of them, are the same as what you were used to when replicating DNA. But it's those rules when it comes to our T's and A's in our DNA strand where things are going to look a little different when it comes to our RNA. So let's look at this process as a whole. So first off, we have our RNA polymerase binding to the promoter region of our DNA, and this is going to do the job of many different enzymes together in that it's going to unwind the DNA while it is producing this transcript. So in prokaryotic cells, the RNA polymerase can bind on its own, but in eukaryotes, we often get accessory transcription factors that bind to the polymerase to create a polymerase complex in order for transcription to occur. Now, RNA is then going to synthesize our RNA molecules by pairing those free RNA nucleotides with the DNA template. It's going to continue to move, unwinding the DNA, building an RNA until it reaches that final region. So we can create several genes simultaneously during the time that we are transcribing our RNA. So if we want multiple copies of a gene, we can have multiple RNA polymerases working at the same time, but they are all going to come to an end when we reach that terminator region. So we're going to have different ways that prokaryotes and eukaryotes terminate the process of transcription. Eukaryotics terminate to form this pre-mRNA molecule. So it does require additional processing before we can go and send it off to do what it needs to do at the ribosome for the process of translation. Prokaryotic RNA is going to be good to go right off the bat. In fact, we can often translate and transcribe at the same time. But we're going to take some time to look a little bit closer at what is involved in this processing to get that eukaryotic pre-mRNA to a state that we can read it and produce a protein. So the first thing that we can do is, with our processing, is we're going to take that pre-mRNA and we are going to make some additions to its structure and splice it up a little bit so that it is ready for the process of translation. The primary events that are conducted during eukaryotic mRNA processing are going to alter the ends of our pre-mRNA. So to the 5' end, we are going to add a 5' cap or a G cap. which is a modified guanine nucleotide. And to the 3' end, we are going to add a poly-A tail, which consists of many adenine nucleotides. And both of these serve very particular uses when it comes to getting our pre-mRNA out of the nucleus. So these help to facilitate the export of mRNA from the nucleus to the cytoplasm, where in eukaryotic cells we are going to translate it. It's also going to help protect our mRNA from being broken down by enzymes within the nucleus. And finally, it helps our ribosomes attach for later translation, or when we go into making those proteins. Now, one of the things that I have not addressed yet is that there may be some elements of this pre-mRNA that need to be cut out. Sometimes we have more information than we need. In our eukaryotic genes, we have regions that are coding and we have regions that are non-coding. So we have specific terms that we refer to these regions. We have exons and we have introns. Now, one of the easiest ways to remember the difference between these two is to think about what they do. Introns interrupt. So they are extra information that we do not need. These are non-coding regions. These are not going to be translated, and so these are what we will cut out of our final product. Exons, on the other hand, are expressed. These are coding regions. They contain information necessary to build our protein. And so these are going to get translated, and so these are going to be spliced or connected together when we form that mature mRNA molecule. Now, one of the lovely things about the eukaryotic genome is that there are sorts of different ways that we can remix these exons together. So we have in this RNA splicing process where we cut out our introns and splice together our exons, and this is all facilitated by what we call a spliceosome. But alternative splicing is this method of us taking different pieces and putting them together in different ways to produce different proteins. So we can have one gene sequence that codes for multiple different types of proteins, all dependent on how we cut and paste those pieces together. So alternative splicing is one of the ways that we can get a wide variety of products from one segment of our genome. And so this increases the potential diversity and complexity that we get within these eukaryotic cells. Now, once our RNA is done processing, it is ready for translation, where we ship it out of the nucleus, we send it over to our ribosome, and we're ready to build some proteins, which is exactly what we'll do in the next video. I'll catch you guys then. Bye.